Hello and welcome to Unit 1.4, Proteins. This is a short revision video. If you would like a longer, more detailed version of this video, then check the top link in the description. You can also find other resources such as flashcards, detailed notes and revision websites in the description. I will now go over the key points of Unit 1.4, Proteins. I will go over enzymes in another video. Let's recap. Proteins have a variety of functions within all living organisms. Amino acids are the monomers from which proteins are made. Amino acids have a central carbon atom to which an amino group, a carboxyl group, a hydrogen atom, and a side chain, R group, are attached. Dipeptides are formed by the condensation of two amino acids. Polypeptides are formed via condensation reactions between the amine group of one amino acid and the carboxyl group of another. A molecule of water is released during reaction. The bonds formed between amino acids are called peptide bonds. The reverse reaction happens during digestion. A functional protein may contain one or more polypeptides. There are four protein structures. Primary. This is a specific sequence of amino acids determined by a specific sequence of DNA bases. Secondary structure. This is a result of protein folding. The secondary structure is held together by hydrogen bonds between the carboxyl groups and the amino groups in the polypeptide backbone. The most common structures are an alpha helix and a beta pleated sheet. Tertiary structure. It is folding into a three-dimensional, globular structure. The tertiary structure is held together by bonds between the R groups of the amino acids in the protein, and so depends on what the sequence of amino acids is, and so is very specific. There are three kinds of bonds involved: hydrogen bonds, which are weak, broken by heat and pH changes. Ionic bonds between R groups. These are broken by pH changes. Disulfide bonds. These are the strongest. They are broken by reducing agents. Quaternary structure. In nature, some proteins are formed from several polypeptides, also known as subunits, and the interaction of these subunits forms the quaternary structure. They contain the same bonds as previously mentioned. Testing for proteins. If you need to find out if a substance, like a food sample, contains protein, you would use the biuret test. The test solution needs to be alkaline, so you first need to add sodium or potassium hydroxide solution. Next you add a few drops of copper sulfate solution. If protein is present, the solution turns purple. If there is no protein, it will remain blue. Chromatography can be used to identify the different amino acids in a sample. Thank you for watching Freeology Revision.